everyone, to the STEAM Club's marker and leaf chromatography session here. And we're so excited to be with you. I am Pat.
And I'm Marcy. And we're going to do some cool chromatography today.
Yes. And let's see, so that you know the way to view the video again, you can go to SCCLD Facebook and to see prior STEAM Club videos like the ones showing up on this slide, you can go to our YouTube channel and search for SCCLD Facebook Kids STEAM Club. All right, so the big question is, what is chromatography? Can you tell us, Marcy?
Sure. As you can kind of see in the photo, chromatography is separating the parts of a mixture so that it can be seen each part by itself.
Awesome. What a pretty picture that is. So that's what you're going to do in these experiments. So chromatography is a valuable technique for biochemists to separate mixtures. So wouldn't they use it for sometimes blood? Is that right?
Sure. It has some medical applications such as separating proteins in blood.
Awesome. And so you can see by using this, you're going to see the different parts of whatever makes up the color. So chroma is color and graphy is tography. I'm not sure if it goes for chromata, but graphy is studying the colors and breakdown of the colors. So.
And you might hear us use the term pigments. So the washable markers we're going to use in our first experiment, they're made up by using different pigments. So what are pigments?
Pigments, substances that make color like ink or dye. And we're using, what are we going to learn about pigments from this experiment? At least the first one. Well, both of them actually, but we're going to see what we're going to use colored markers first, but we'll figure that out in just a minute. But we're going to see what pigments make up each color in first markers and then leaves. Mm-hmm. So.
And one of the ways we're going to do that is we're going to use water as a solvent for the first experiment. So they're washable markers, and so the water can kind of wash through the colors, and we'll see what happens.
Excellent. So here are the things that you need for our experiment. So we've got the washable markers, like Marcy said. We're going to use paper towels, cut into 1 by 5 inch strips, and scissors to cut the paper towels into 1 by 5 inch strips. Clear glasses, something to pour water with, pencils, and tape. And Marcy has a good thing to say about these strips if you don't have them exactly the height of 1 inch by 5 inches.
Right, that's just an approximation. So when we were doing our experiment, we found that we were using a glass that was pretty thin at the bottom, pretty narrow. So we had to cut off a little of the 1 inch to make it work. And we're going to use the pencils to roll up the ends so that the papers will fit in the glasses properly. So don't worry if this is just an estimation of what to cut the paper towels in, and we'll go from there.
Sounds good.
But we're scientists, and scientists always make a prediction before they get started. And so this is just an example, is what color pigments make up green? So Pat, what do you think? What would be your best prediction, estimate, hypothesis. What colors do you think will make up the green?
So I'm just remembering from what I learned a long time ago. I think green, you get green when you mix yellow and blue. So I'm going to predict that.
So for each color, you can make a prediction. You could write it down, or you can just verbally say it out so you remember when you get to the results to see how close you were.
Yeah, that makes it fun. Okay, so you're going to pick out the different markers, and it's really nice if you pick out as many colors as you can because it's really fun to just experiment. So here, and then we picked out blue, red, purple, green, brown, and black. And we drew thick lines of the markers on the paper towel about 1 inch up from the bottom. And we also wrote at the top of the strips. What did we write up there, and why did we write that?
We wrote the colors because when we were practicing this, we thought that there was a particular marker that was used, but then when we looked at the top and saw what the color was, we were wrong. So you think that you could think that you know what marker it is, but to be sure, write it on top before you even get started.
Excellent. And wow. Yeah, this is an important one. Before putting the water into the glass, first make sure that you've got the strip the right length so it just touches the bottom of the glass. So do you see how you wrapped it around the pencil, and then you can tape it right when it's the right length for just touching the bottom of the strip to the water.
So the next step, we want to take the strip out before we add the water because we might splash water on the strip. So if you put a little water on the bottom and then you put your strip back in, then it's just going to be the bottom that gets wet. We want only a little water. We don't want it to go all the way up to where the marker is because it would wash out the marker before we even get started.
So once you put the strip back in, and Marcy pointed out to me when we were doing this that you want to keep the strip kind of free from the sides of the glass, in addition to just barely touching the water. And you can watch the water creeping up the strip. So this is the green, right?
This is black. I did
black.
mark on the bottom
right there.
Colors.
Okay. Yeah.
You may get different results, but we wanted to show you what colors we were using. So even with this being in touching the water just for a couple of seconds, it already starts looking like there's green in the black marker.
Right.
And what's happening is the water is being absorbed kind of like a sponge up the paper towel. And so the water's just going to keep going up and up.
Right. So you want to remove the strip within 10 minutes, and you can dump the water out and put it into that, sort of dry the glass out and put it into the dry glass so that it dries out. And that one looks like it might have been purple. Yes, it is purple. I need to read your slides. Marcy made these beautiful slides. Okay, so
I had one that I left too long. This one with the markers, we don't keep it in for very long. So what happens if you leave it in for too long?
What happens? Look,
Yeah.
There it is. Look what happens.
The ink in the water just seeped right up to the top, and now you can't even see what the results were. It was blue, but you can't even see if it was just blue or made up of different colors.
So it got super saturated. Okay. So what pigments did each color break down into? So this is blue. Blue it looks like purple and pink, sort of purple, right? Or pink?
Uh-huh.
Pinky purple.
That was a surprising result to me. I predicted blue would just be blue.
Yeah. And it is kind of, right, it's very dense at the top, but then it's lighter and then it's purpley.
Mm-hmm.
That is cool.
And so then you can compare. Are there any similarities between the strips? Are there one particular color that comes up to the top? These are both samples of black using two different kinds of markers. And there's some similarities. I see a little pink on the bottom of that, and I see the blue going up to the top. The second marker on the right does not look like it had any yellow.
Right. Yeah. I just think it's amazing that these two colors have so many colors in them, right?
Mm-hmm.
And they're both black.
Right.
Yeah. So that's pretty interesting.
So let's see.
So chromatography, separating the parts of a marker so that you can see the pigments one at a time. And look at that. What do you, so yellow at the top, it's, as a scientist, you can compare whether the other strips had yellow at the top, or is that the one that gets absorbed fastest? Is that what we're saying? Is that
so how fast each pigment travels depends on the pigment's molecules and how strongly the pigment is attracted to the paper.
Okay, that's interesting. So it looks like, at least for a couple of these, maybe for the black ones at least, the yellow got absorbed fastest.
Yeah.
Yellow, green, and purple. Wow. And purple was the slowest.
Oops. So what other things can you do? This is just the beginning. If you just tried out a few markers, try out a whole bunch of different colors because you may be surprised what they're made of. Use 3 of the same color, but different kinds of brands, and see if you come up with different results. Black works really well because, as you saw, black is made up with lots of different colors, and so you might find lots of fun results with that. You could even use different kinds of paper. Pat, what kind of paper do you think we could use other than the paper towels we used?
So you could use coffee filters, toilet paper. You could use regular paper. You could use tissue paper, not colored, like, what is it? Tissue that you use for your nose and your facial tissue. But yeah, you could just use a plain sheet of white paper too, and you can just see what happens with those and which ones work the best at absorbing and finding out what color breakdown is.
So you could make a hypothesis. What's the best type of paper to use? And think about how the water would be absorbed by the paper. I think that's going to be one of the keys. If you really wanted to have fun, you could use a couple different markers, either put 2 different lines and then see how the water absorbs through them, or you could make even a little picture or something. We just want to have fun with science.
Yeah. I'd like to, I hope some of you do try it to mix or do a picture with different colors and see how that affects the paper. So
now we're going to continue what we learned from this marker experiment. We're going to do with leaves.
Okay, so leaf chromatography, yay! So I guess the question is, what does a tree need to grow? What do you think a tree needs to grow, Marcy?
Well, I see in this picture, sun.
Sun.
And I know that the roots are in the ground to get water.
Right.
And nutrition from the soil.
Right.
And then leaves also need to breathe in the carbon dioxide. We breathe in oxygen, but plants, trees need carbon dioxide. So 4 different things: the sun, water, nutrition from the ground or soil, and the carbon dioxide are 4 main things.
And that kind of goes with, I don't know if you've ever heard that you're supposed to talk to your plants. Well, it's actually, you don't have to, but it is nice because the carbon dioxide comes straight out of our mouths and goes to the trees and the plants. So
here's a little diagram that helps us see that. There's some people in the bottom there, and they must be talking to the tree because they're giving it the carbon dioxide and the tree's giving it oxygen.
That's right. And they gives it oxygen, the sunlight comes in, and water from the soil, and this process called photosynthesis, all these things contribute to the process of photosynthesis, which produces sugar and oxygen and something else very important there. It's, what is it, Marcy?
What is the green you see in the leaves? That's the chlorophyll.
Chlorophyll.
And chlorophyll is the most common pigment for leaves, and photosynthesis produces chlorophyll.
Right. So that's what makes the leaves green. Very nice. Look at that. Look at that. That was interesting. Sorry. Pigment.
I think I went 2 minutes. Okay.
Oh, sorry. Okay, so here we see pigments again. We're talking about pigments, and it's the coloring matter in living things. So chlorophyll is one of the pigments, right? And what other colors are in this? Red and orange and black.
Yellow. Brown.
Yeah. And do you see the veins inside these? Well, we'll talk about that in a minute, because materials for leaf chromatography. So what is the big difference here? We're going to use rubbing alcohol rather than water. And you're going to use leaves and get a lot of different kinds of leaves, the green ones, and make sure to get some maple leaves that maybe have different colors in them. And you got the paper strips. A lot of them are the same things as you needed from before, but there's a pencil in there, but what is the other pencil for, Marcy?
So one of the pencils has not been sharpened, and we're going to use that kind of as a tool. But if you don't have a pencil that hasn't been sharpened, you can use the eraser end as our tool later on. But yeah, if you can find an unsharpened pencil, that would be great. And the reason we're using rubbing alcohol instead of water is rubbing alcohol is going to be our solvent. Remember last time water kind of broke down the washable markers? Well, now we're going to use the rubbing alcohol to be a solvent. Water may not be strong enough. We may need a different material. As scientists, you can always try this experiment with water and see if that works. But to be soluble means to how much the substance can be dissolved. So we're going to try to get some of that chlorophyll out of the leaf and other colors. And so that's why we're using rubbing alcohol.
Cool. And then the other thing that if you happen to have one at home, a magnifying glass, you could use that. It's optional, but you could use that thank you. This is great. So you could use that to study your leaves, the veins of the leaves. Do you see all those veins? And that helps you look at it more closely before you start looking at doing the chromatography experiments.
And again, we want you to make that prediction with whatever color leaf you have. Make your prediction. What colors are going to appear on the paper filter when we're through?
Great. So cut up leaves. So the first step is you can tear the leaves up, but Marcy discovered that it was really helpful to use those scissors to cut the leaves up because it gets a little tedious to try to tear them up. So do whatever you feel best, but do a lot of leaves for each separate glass container.
So are you going to cut all the leaves that are this different kinds of leaves and put them up there, or what's your strategy? What was your strategy?
Mine?
Yeah.
Oh, well, I had a big maple leaf and it had different colors in it. So part of it was greener and part of it was yellow. It had already started to change colors because I guess it was getting less chlorophyll. So I took some of the green from it and tore that up. And then I took some of the brown and yellow and tore that up. And I tried to keep samples of each next to the glass that they were next to so that I would remember which type of leaves were in the glass.
So, yeah. Oh, sorry.
No, go ahead. Go ahead.
So the strategy is to keep the same type of leaves together and then cut them up and put them in. And I also saved, just like Pat did, I saved a leaf from each one that I was cutting up. So I would remember what kind of leaf sample it was in that cup. So we used different glasses for the different types of leaves.
Right. And then, yeah, so this one, Marcy when I was doing it, I didn't pour enough rubbing alcohol over, but it was really helpful because even in just getting this experiment together, we needed to experiment to get best practices for you. So Marcy figured out that you want to pour the rubbing alcohol over the leaves and have them at least covered by the rubbing alcohol, enough so that they're covered. And then you're going to use this is the part Marcy discovered. So you tell them about this.
Sure. So we use that unsharpened pencil kind of to mash the leaves. You can see the rubbing alcohol is still clear. And it takes time for the chlorophyll to come out of the leaf. And so we mashed it to kind of try to see if that would help release it. We also left it for a longer period of time. We left it for at least an hour, and it's even better to leave it overnight before you do the next step.
So that's really interesting. So before you even put the paper in, is that right? Okay, that's cool.
And so you can see now this has been left there for a couple hours, and so the liquid is greenish. And if you didn't have enough liquid for when you put your paper towel in for it to absorb anything, you could add some more rubbing alcohol now and just kind of stir it up so that it has some liquid to absorb.
Right. And just like Marcy said, make sure you stir it so that it gets that color in there. And now you're going to put the strip in the glass so that it's just barely touching the liquid, and you can wrap it around the pencil and just tape it onto the pencil. And see it's starting to absorb there. Now I wonder, this is one of the big differences here, Marcy, is do you take it out after 10 minutes?
No. So again, it's going to take a lot longer. This experiment, you need to keep it in for at least 90 minutes. And again, if you could just keep this overnight with the paper in it, you're going to get a lot better results.
That's cool. So that's good to know. So look at this. You can watch as the rubbing alcohol creeps up the strips, and you don't have to empty. Keep the rubbing alcohol in until you're completely done. And when you are, then you can take it out to dry. But isn't that interesting? The top one. What was that? I can't even remember what the leaf was.
I'm not sure what leaf that was. We didn't do as good a documentation as we should have. As scientists, that's one of the important things is to document. And that one I don't have.
Well, that's okay. It's pretty though, isn't it?
But the one below is the one that was your huge maple leaf.
Yeah.
And it did have different results based on what part of the leaf you used.
Right. Yeah, so the one on the left is the greener part, and the one on the right was the part that didn't have much green in it and more brown. So, yeah.
Another thing we saw that were some of the leaves were drier than the others. So those are the ones that had to soak longer as well. They had to kind of get over being dry and start absorbing that rubbing alcohol.
Yeah. Yeah. So look at this. Do different colors appear, and did the same colors always appear on top? So look at these. These are all green leaves. And it looks like, well, one of the middle one definitely has like a brownish color on top.
And that was spinach. That was some spinach from my my garden that we used on that one.
Right. That was really strong. Down at the bottom, the green was really strong. And the other ones don't have, the one on the right, the thin green one has a little bit of brown, sort of a brownish on top. And the other one over on the left looks sort of whitish, sort of very light green to white. So it's really interesting.
They have some similar results, but yeah.
Yeah, definitely very similar.
So make sure you choose a lot of different kinds of leaves and you'll get a lot of different results. Like we found a kind of that purple leaf, and we were able to have that in our experiment too.
Right. Oh yeah, and I also one of the things I noticed was how intense the green was from the spinach leaf. And just remember, if you're a hungry person like me, don't eat any of the leaves, even if they're spinach leaves, because you don't eat any part of anything that you're using in an experiment. So
you're just recommending during the experiment and the materials you're using at the experiment, but you would eat spinach, regular spinach, in your salad not from the experiment, but you would eat it anyway.
Right. Only
on a regular basis.
Just not in the experiment.
Right.
Wow.
So we do have books.
Yeah,
if you like this and you wanted to explore more about why leaves change colors, the library does have a collection of books that you could go to the library and get.
Yeah, these are really good books Marcy found. And since you can view this again you can view this
video,
thank you. Again, you can copy down some of these names of the titles, but these look like really good books. And they'll help you as you do more experiments. So
we just wanted to remind you that the STEAM Club and the Art Club will be doing more programs on the first Tuesday of every month. It's every other month will be the STEAM Club, and then every other month will be the Art Club. So make sure to join us on Tuesdays, the first Tuesday of the month at 4:00.
Thank you so much, Marcy, and thanks to everybody for joining us.
All right. We'll keep the experiments going. Thank you.
Bye.
Bye.